Hi, everyone. In this presentation of the special symposium on multimodal imaging, we will analyze how we can actually investigate the coupling between structure and function with two different modalities, in this case of magnetic resonance imaging, but with methods that can be also applied to other type of functional imaging, for example, MEG or EEG. In particular, I will guide you through a recap on how we can quantify brain structure-function coupling, so by decomposing the functional signal from fMRI with structural harmonics, so based on the structural connectome from diffusion tensor imaging. But also, I would like to focus your attention on how we can investigate temporal dynamics and the preliminary results of our most recent research on that. Okay. The two pieces of information that we're going to use are acquired with magnetic resonance imaging. The first one is the functional information about brain activations from fMRI. And the second one is the structural information given by the virtual reconstruction of white matter bundles physically linking different regions of the brain. Obtained by diffusion MRI. So combining these two pieces of information is not trivial, and we do it today with graph signal processing. In particular, also we know that brain function is dynamic, so we also want to explore how does the structural function coupling varies over time. What we consider as graph is the structural connectome that is also characterized by an adjacency matrix A and a degree D. And what we consider as graph signal is a functional activity frame at one time point. So a vector that attributes a value to each of the nodes of the graph. Also, this signal will vary across time, and we will apply the same pipeline to each time point. We want to explore how is the function using the underlying structure. Quickly, this is a pipeline that we developed, and the first step is the decomposition of the structural connectome into a set of basis functions. These basis functions are obtained as the eigenvectors of the Laplacian of the structural connectome, also called structural harmonics, because each is associated to a value of spatial frequency, which is given by the eigenvalue lambda. You can see that the eigenvalues go from 0 and then increase. So the low frequency, the first eigenvectors will have low frequency, and this means that we will be smoother on the graph, more coupled to the underlying graph, while the further eigenvectors will have high frequencies, and so a higher level of a lower smoothness on the graph, so more detached from the underlying structure. The second step corresponds to decomposing then the functional signal, so the fMRI frame at one time point, over this structural harmonics. This is equivalent of performing graph Fourier transform by estimating the spectral coefficients as hat that encode the amount of each structural harmonic included in the fMRI frame at one time point. Then we perform a filtering, low pass and high pass, to reconstruct two portions of the original signal, one that is only contains low frequencies that we identify as a coupled portion of the signal, and the other one with only high frequency that is more liberal with respect to the structure. If we repeat that for each time point, we obtain a coupled and decoupled version of the time courses, and then we computed the L2 norm across time of the coupled and decoupled time courses, and then the ratio of these two quantities, so decoupling versus coupling, is what we define as structural decoupling index, a node-wise metric of structural function coupling. And then similarly to what we do when we compute functional connectivity from normal time courses using Pearson correlation, we apply that to coupled and decoupled time courses to obtain two versions of the functional connectivity, one coupled and one decoupled. These are therefore the final metrics that we're going to use: node-wise a structural decoupling index, and edge-wise coupled and decoupled functional connectivity. But we want to look at variations of these metrics. So we apply this pipeline in a sliding window framework here, applied to 100 LC controls of the HCP database during different resting states and with two different windows for which we obtain very equivalent results. So the results are presented here for the longer one. So in a sliding window framework, we computed SDI, coupled FC, and decoupled FC, and then we concatenated, after vectorization, all this data into a big time per subject matrix. Also, after the important operation of temporal demeaning that allows us to look at variations from the mean pattern of this matrix. Then principal component analysis allows us to obtain what we call eigenpatterns of dynamic structure function coupling that encode the variations from the mean, from the static pattern. You can see these in variations here encoded with red and blue colors, so increases or decreases from the static pattern. These are the first 6 components of the SDI decomposition, and what we can notice is that the first global component has the largest variance, so meaning that for a considerable amount of time, indeed, the average, what we call the static pattern of SDI, is dominating. But then further eigenvectors, they give us information about, they enrich the view about the temporal variations of this pattern, and they represent, again, global patterns, low frequency dominated and reminiscent of resting state networks. As you can see here, for example, default mode network opposing to sensory motor and visual network, or here frontoparietal and temporal against, in blue, sensory motor network. So this is not trivial, since again, we are looking at variations of the signal from the mean. A similar situation occurs when we look at the coupled functional connectivity decomposition, while a different case we have with the decoupled version, where the variance is equally shared, meaning that we have more temporal variations across time, and also these patterns are more localized. They encode high frequency components, even if we can notice that they're still symmetric left-right. So in conclusion, here we explore for the first time the dynamic eigenpatterns of structural function coupling. The ones of SDI and coupled FC involve non-resting state networks, while the decoupling pattern of functional connectivity showed to be more variable across time, and this is consistent with the higher value for fingerprinting that was also previously shown in our previous work. Future study will further explore the physiological and clinical relevance of this pattern. For example, back projection would allow for the computation of temporal occurrences that would be useful, for example, in task-based setting. And with that, I thank you for your attention and thank my co-authors.